neoplasia. And first of all, we will discuss basic principles in the topic of the neoplasia. So the neoplasia is actually the neoplasia means the new abnormal tissue growth that is unregulated, irreversible, and the monoclonal. These three features help differentiate it from the hyperplasia and from the regeneration. As you know that in hyperplasia there is a controlled increase in the cell number, and in the repair or the regeneration there is a normal healing response. The plastic cells continue to grow even after the original stimulus is removed, and the growth occurs due to the accumulation of the genetic mutations affecting the cell cycle regulation. Monoclonality. It means that the monoclonal means that the tumor cell arises from a single mother cell, and all the neoplastic cells therefore share the similar genetic abnormalities. Benign and the malignant tumors are usually monoclonal in origin. Determination of the clonality are the glucose 6-phosphate dehydrogenase isoenzymes. Clonality was historically determined using the glucose 6-phosphate dehydrogenase isoenzymes. Number first concept is that the multiple isoforms exist. That is the glucose 6-phosphate dehydrogenase A and the glucose 6-phosphate dehydrogenase B and the glucose 6-phosphate dehydrogenase C. The second concept is that the one isoform is inherited from each parent. In females, one X chromosome is randomly inactivated in each cell, which is called as the lyonization. Normal tissue show a 1:1 ratio of the isoforms. For example, 50% of the cells of the glucose 6-phosphate dehydrogenase is A, and the 50% cell is the glucose 6-phosphate dehydrogenase B. Hyperplasia is the polyclonal, so 1:1 ratio is maintained. And the neoplasia is the monoclonal, therefore only one isoform is present. Clonality can also be assessed using the androgen receptor isoforms, and these are also X-linked markers. Monoclonality supports the idea that tumor originates from one transformed cell that undergoes the repeated proliferation. Now we'll talk about the tumor nomenclature. Tumor naming depends on the lineage of the differentiation and whether the tumor is the benign or the malignant. So the table given, in this table we will talk about the examples of the tumor nomenclature. So the first thing is the lineage, and the second is the benign, and the tumor or the third is the malignant tumor. So the epithelium, if the lineage is of the epithelium, so this benign condition will be known as the adenoma, and the malignant condition of the epithelium will be the adenocarcinoma. And in the epithelium, benign may be the papilloma, or the malignant may be the papillary carcinoma. And the next is the mesenchyme. The benign condition of the mesenchyme is known as the lipoma, and the malignant is the liposarcoma. The next is the lymphocytes. Lymphocytes are almost the malignant, or the lymphoma or the leukemia. Melanocytes are the benign condition is the mole or the nevus, and the malignant is the melanoma. Malignant epithelial tumors are called the carcinoma, and the malignant mesenchymal tumors are called the sarcomas. So these are the simple concepts from the neoplasia. Now we'll talk about the common screening methods. The first one is the pap smear, and then is the mammography, and then is the PSA, means the prostate specific antigen plus the digital rectal examination, and then is the hemoccult test plus colonoscopy. So now we'll talk about the pap smear in detail. Pap smear detects the cervical dysplasia or the cervical intraepithelial neoplasia. It identifies the precancerous lesion before the invasive carcinoma develops. The microscopic finding in the pap smear is the dysplastic squamous epithelial cells with a nuclear atypia. The next screening method is the mammography. Mammography detects in situ breast cancer, the ductal carcinoma in situ in case of the breast cancer. It helps identify the lesion before they become palpable. The gross concept in this is that it may show microcalcifications. The third screening method is the PSA, or the prostate specific antigen plus the digital rectal examination. This is used for the prostate carcinoma screening. So PSA, or the prostate specific antigen, may also increase in the benign prostatic hyperplasia or the prostatitis. The next is the hemoccult test plus colonoscopy. It detects occult blood in the stool, and colonoscopy identifies the colonic adenomas and the early colorectal carcinomas before the spread occurs. The gross morphology in case of the hemoccult test or the colonoscopy is that the adenomatous polyps may appear as the raised mucosal lesions, and the macroscopic feature in this case is the dysplastic glandular epithelium with a nuclear hyperchromasia.